Hi there, I'm Shin Hatou, CEO of our company Cellusion. Cellusion is a startup company from Keio University School of Medicine. My background is ophthalmology, and my specialty is corneal regenerative medicine. Today, I would like to introduce our project, corneal endothelial regenerative medicine with iPS cell-derived endothelial cells. There are unmet needs in corneal transplants. It is reported that 30 million patients are waiting for corneal transplants globally. However, only 180,000 corneal transplants are conducted per year globally. So the demand is 70 times the supply. On the top, you can see the clouded cornea. This is bullous keratopathy, corneal endothelial cell dysfunction. Bullous keratopathy is the most common disease for corneal transplants. More than 50% of corneal transplants are for this disease. Bullous keratopathy is caused by corneal endothelial cell loss due to the surgical complications such as cataract surgery or genetic abnormality such as Fuchs corneal dystrophy. Current treatment for bullous keratopathy are penetrating keratoplasty or endothelial keratoplasty. Although surgical technique has been advanced, there still be many complications and they are still very difficult surgeries. So what about our proposed solution? Our solution is corneal endothelial substitute cells from allogenic iPS cells called CLS1 cells. We can produce corneal endothelial cells from allogenic iPS cells with mass production, standardized quality, and stable supply. Also, we have developed cell transplant technique. Suspended CLS1 cells are injected into the cavity between cornea and lens called anterior chamber space. This surgery is very low invasiveness, easy of handling, and ready to use care. Here you can see the CLS1 preclinical efficacy study with monkey bullous keratopathy model. Top left, you can see the corneal edematous thickness from baseline. CLS1 transplanted eyes in blue significantly reduced its corneal edematous thickness compared to the negative control eyes without cells. At the same time, CLS1 transplanted eyes dramatically recovered its transparency compared to the negative control eyes. Also, we have confirmed that the transplanted CLS1 cells covered the posterior surface of the cornea. So, CLS1 transplants reduced corneal edema, recovered corneal thickness, and recovered corneal transparency dramatically. And how about the production process and characteristics of CLS1? Left, you can see the production process of CLS1. Frozen stocks of iPS cells are thawed, cultured, and expanded. It takes about 3 weeks for expansion culture of iPS cells. After that, CLS1 endothelial cells are differentiated from expanded iPS cells with attached culture. It takes for just 2 weeks. Finally, CLS1 endothelial cells are preserved as frozen stocks. The cells are preserved until cells are used. So right, you can see the immunostaining of corneal endothelial cell markers of CLS1 cells. CLS1 cells are quite similar to the true corneal endothelial cells because CLS1 cells express NAK-ATPase, ZO1, N-cadherin, and transcription factor PITX2. And also you can see the flow cytometry analysis of functional corneal endothelial cell markers. you can see that the CLS1 cells express N-cadherin as barrier function, NAK-ATPase and mineralocorticoid receptor as pump function, with quite high positive rate. Okay, how about the purity of CLS1? Top, you can see the immunostaining of undifferentiated cell marker OCT4, OCT4. iPS cells express OCT4, whereas CLS1 cells don't express OCT4. And bottom, you can see the undifferentiated cell population by OCT4 qPCR analysis, which reveals that the undifferentiated cell population in CLS1 cells is below 0.01%, which is represented by the red dotted line. And also, you can see the cell growth analysis of CLS1 cells, and which reveals that there are no cells that proliferate abnormally in CLS1 cells. Okay, here you can see the brief summary of CLS1 preclinical safety studies. We have also conducted in vivo tumor genicity test, which is cell transplants into animal anterior chamber of the eye. This method is also patented by us. And we have confirmed that there were no tumor genicity nor teratoma formation of CLS1 cells. Also, we have conducted residual rate of undifferentiated cells, genetic chromosomal abnormalities, general toxicology, and pharmacokinetics, and we have confirmed that there are no safety concerns in preclinical studies. Okay, how about the CLS1 targets? CLS1 targets mild or moderate vasculopathy patients as first-line therapy because of its CLS1 injection therapy is very low invasiveness and far easier compared to the conventional keratoplasty. And here you can see the development plan of CLS1. There are 3 points we would like to highlight. First, investigator-initiated clinical study at Keio University Hospital has been started this year. This is the first in-human clinical study. After that, Cellusion-initiated phase 1-2 clinical trial in Japan is scheduled in 2023. And in the US, phase 1-2 clinical trial is scheduled in 2025. So we have continued the PMDA consulting, and we will start the FDA consulting this year. Okay, how about the competitive advantages and IPs? We can produce CLS1 endothelial cells from allogenic iPS cells in just 1 month by our unique direct differentiation induction method. Secondly, we can treat more than 1,000 patients per batch. And thirdly, we can preserve the CLS1 cells as frozen stocks, which allows us to distribute the cells to the medical institutions all over the world. These advantages are totally protected by 7 patents. Okay, how about the competitive landscape? We are the pioneer of iPS cell-based cell therapy for vasculopathy. Although there are some competitors who use cultured corneal endothelial cells from donor cornea, however, the cell number from donor cornea is very limited, and the cell quality varies due to the donor variation. By contrast, we can mass produce CLS1 cells with stable quality. And also, we can preserve the endothelial cells, as I mentioned before. So how about the business model of us? Cellusion produced the CLS1 endothelial cells with CDMO. and distributes the cells to common medical institutions all over the world. General ophthalmologists will be able to treat pleural patients all over the world with our cells, CLS1 cells. And its cost will be acceptable for medical insurer because of the mass production and the preservation. Okay, how about the annual new patients forecast? The patients' number of pleural are forecast to steadily grow around 1.2 million per year, which translates into market size as $5.8 billion per year annually. It is due to the growing country's medical level, increase of intraocular surgery and complications, and population of aging. Okay, how about the launch schedule? We will launch CLS1 cells in Japan in 2026, in US in 2029, and the rest of the world in 2030. Finally, I would like to introduce our management team. Again, I'm Shin, CEO of our company. Georgie Hayashida as COO, formerly at Astellas Pharma. Michihiro Hayashi as CAO, formerly at MBL and IDC Therapeutics, and he has experienced the IPO. And Nobuyuki Imai as CDO, formerly at Kyowa Kirin. Okay, this is take-home messages. CLS1 is a leading program of iPS cell-derived corneal endothelial regenerative therapy and preservable off-the-shelf cell product with stable quality and a mass-producible cell product to fulfill the global pleural patients' needs. Okay, that's all. Thank you for your kind attention. If you are interested in us, please contact here or website. Thank you very much.